So, welcome back to the Purdue University Rosen Center for Advanced Computing Cyberinfrastructure Symposium. I'm Lisa Arafine, and I'm a partner with the Rosen Center. Some housekeeping items to make note of. Please silence your phones. Please ask questions at the end of talks. Please join us at the conclusion of the CI Symposium today at a reception in the Purdue Union East Terrace from 4:15 to 5:30. Everyone is welcome to join. The East Terrace is accessible from the Union basement level and is located where Pappy's Sweet Shop used to be. Okay, now I would like to welcome Ananth Grama. Dr. Grama is the Samuel Conte Professor of Computer Science at Purdue University. His research interests include parallel and distributed computing, large-scale data analytics, and applications. He is a fellow of the American Association for the Advancement of Science, distinguished alumnus of the University of Minnesota, recipient of the National Science Foundation Career Award, and the Purdue University Faculty Scholar Award. He received his bachelor's in engineering from the Indian Institute of Technology, Roorkee, his master's from Wayne State University, and his PhD from the University of Minnesota. And he's been at Purdue since. Today, he will talk about computational methods for analyses of functional brain connectomes. Welcome, Ananth. See the controller? Okay.
Yeah.
Great.
Just make sure it works. Thanks, Lisa. Thank you for having me here. It is a pleasure, and I do apologize. This was not the topic of the talk that I was going to give, but that was going to be some really, really cool stuff which we were building with people, the good folks at Rosen, but we were not clear to talk about that, and so I won't be talking about that. Instead, I'll talk about something else we've been working on, which we think you'll find fun and exciting. I'll keep this talk at the level more of applications, but there's a lot of math and algorithms in there, and I'm happy to engage at any depth on any of those issues. All right, so this is recent work that we've been doing in our group on analysis of MRIs. Basically, how much can you learn from MRIs? Really, really cool stuff. And most of it is work done by a PhD student of mine, Vikram, who just graduated, who is at the University of Cincinnati himself at this point. Some of the results also are due to joint work with another PhD student of mine, Chi Hao, and a couple of colleagues, David and Petrus. All right. I assume we are a relatively heterogeneous crowd, so I'll just give an introduction to what is brain connectomics, and then I'll dive straight into the three problems I'll talk about today. The first one on finding signatures in connectomes, the second one on predicting canonical response, and the third one on reconstructing visual stimulus. The way to think about this is fingerprints are unique aspects of our brain, and what I'll show you is that I can actually find out, I can tell you exactly what defines you in your brain. So I will tell you roughly about 60 to 80 features in your brain that nobody else has for the most part. It's yours and yours alone. So those are attributes. The next part, I'll look at a bunch of people who are watching videos. In fact, the first episode of the BBC show Sherlock, I'm sure we've all seen it. And basically, I'll show you that I can extract things from the MRIs that are strongly correlated with what you're seeing on the screen. So that's kind of cool. So, I'll show you not only can I extract that, these are also predictive. So if you give me a set of features, I can tell you a little bit about what you're probably watching. And the third topic is really, really cool. There, I'm truly reading your brain. So these are people who are sitting in MRI machines watching videos. And if you give me the MRIs, I'll tell you what they're watching. Of course, this is very, very coarse, but it's almost scary that you can even do any of this at all. So that's almost unpublished. It's very recent work, but really, really cool stuff. And you can imagine you can do very cool stuff with it. So we're actually talking to Procter & Gamble, where they're interested in sleep studies. So they want to sell you sleep supplements. So can we look at people's, their interest is more in low-cost imaging modalities like EEGs, not quite fMRIs, but nevertheless, can you predict sleep states from EEGs? We're talking to another company, a big German automobile manufacturer. We haven't signed all the contracts, so I can't tell you which one, but you can guess. And these people are looking to put sensors in the back of the headrest to assess driver state. And so our role in that project is to actually get that data and actually say something about driver state. So it's really cool stuff, fun stuff. But we'll start simple here. So let's start with the topic at hand, which is connectomics. So what is a connectome? A connectome is basically a map of the brain. You can have structural connectomes, you can have functional connectomes. So structural connectomes would be, sorry, there's not a pointer in this. Structural connectomes would be the structural maps of the brain. So for example, different parts of the brain and how they're physiologically connected to each other using fiber tracts, for example. Functional connectomes of the brains are basically monitoring electric activity in your signals in your brain, where, for example, if you're doing something, which parts of the brain fire at the same time. So we're talking about correlations in the time series that correspond to the electrical signals that are monitored from the brain. So think of these as your brain connectome is translating to a matrix where the high correlation regions in that matrix are two parts of the brain that fire consistently together while performing a certain task. So you can think of these as task-specific MRIs. You can think of these as resting state MRIs. For the most part in this talk, I will only talk about functional connectomes, but a lot of what we talk about can be correlated back to structural connectomes as well. All right. Just by way of terminology, structural connectomes are sometimes called wiring diagrams as well, and functional connectomes, basically, as I mentioned, measure coherence in neuronal firing at rest or while performing tasks. A few more pre-processing tasks. It turns out that this takes a little bit of a startup to start working in this area. But there's really nice software as well. So you don't have to start at ground zero. You can download a lot of stuff, which people have done a really good job putting together. So your starting point is a noisy functional image. The first thing you want to do is distortion correction. Nobody sits absolutely still in an MRI machine, so you have to do head motion correction next. We really don't care about the skull, so we strip off the skull next. Then you have to normalize and register because everybody's brain is not the same shape or size. So you have to register with respect to a standard coordinate brain, which is called MNI. So we stretch and shrink people's brains to the point of fitting them on a common coordinate system. And that's the normalization and registration part. And finally, you're to a point where you have a sequence of images that you can do something with, which is where a lot of what I'm going to talk about. So a lot of the pre-processing is canonical pipelines. We don't necessarily have much to do with that. Once you pre-process the brain, the functional image, the process functional image is where our innovation comes up. All right, so let's look at the very first problem, which is finding signatures in connectomes. So what are signatures? Signatures are signals that code for individual level uniqueness. As I mentioned, these are your fingerprints. These are things that are absolutely unique to you. The first question, obviously, is do fingerprints actually exist? And indeed, there's work that predates us by about 5 to 10 years, which shows that fingerprints do actually exist. And this is wonderful work in terms of proof of concept, but conceptually, all it is doing is that it'll take your functional activity, and then it'll compute the correlation with your own functional activity later. And what it says is that you are more strongly correlated with your own activity in the future than anybody else. Basically, what that tells you is that there is a fingerprint. It doesn't tell you what the fingerprint is. It doesn't tell you physiologically what parts of the brain your fingerprint will be in. But statistically, it tells you that your response to a stimulus is more similar to your own than to anybody else, which is a wonderful starting point for us. All right. So the experiment is set up as follows. On day one, on machine one, there's a whole bunch of people who all had their MRIs. Now, 6 months later, on some other machine, there's a whole bunch more MRIs that you've taken. And the first question to ask is, can we find corresponding entries can we find your specific MRI on day K on machine 2 if I were to give you your MRI on day 1 on some other machine? And like I mentioned, people have answered this question in the positive, that yes, we can do this. Now, the question is, can we find the markers? In other words, what makes you unique? Not that you're unique. Of course you're unique. We know you're unique. But the question you're asking is, can we find markers that are unique for identifying individuals? Can we find markers that are associated with tasks? So if you were, for example, doing emotional processing as opposed to gambling, do different parts of the brain light up for you as opposed to some other tasks? So can we find markers that identify tasks being performed by subjects? Can we find out how well you are doing at these tasks? So for example, if you were gambling, could we tell whether you were winning or losing from what we see in your brain? Can we find signatures exclusively in the functional connectome as opposed to the structural connectome? And the answer to all of these is yes, and I'll show you how we can do all of these things. All right, as I mentioned, methods that predate us do not prescribe spatially localized markers. They don't even tell you what the markers are. They just tell you that, yes, fingerprints exist. And our goal in the first part of this talk is to find localized regions of the brain that code for the identity. And the mathematical tool for us is something called leverage score sampling. I'll briefly give you a sense of what leverage score sampling is, but I won't go into any great detail. But the idea here is that we use leverage scores of connectomes on day one to select features that are strongly connected, strongly correlated in the corresponding image on day k. Okay, so I have to find those features first. So roughly, here's the setup. I'm going to take all of the subjects. I'm going to take all of their connectomes. I'm going to vectorize them, and I'm going to do the same thing for task 1 and task 2. I'm going to find the markers for these tasks from these. And the markers, just think of markers as something that's unique to these tasks. That's how we're defining markers for now. But mathematically, these are rows in that matrix that have the highest leverage scores. Okay, so that's how mathematically we define these things. So once I've defined those markers, then I only compute correlation in that marker space as opposed to the total space of those vectors, which has got lots of noise, and you will never find signal. So once you pull those markers out, you can find the correlation. Out comes the correlation matrix, and you can do fun things with that correlation matrix. Just to give you a quick sense of what this mathematical doodad is called leverage score sampling, if I give you an n by d matrix A, what I'm asking you is give me a subset of this matrix. So pull out some rows or columns of this matrix, such that if you're so the way to think about a matrix is what are the distance between rows or columns of this matrix. So if you think of the matrix A transpose, the ijth entry is the dot product of the ith row and the jth column, which means that that's the distance or the correlation between the ith row and the jth row because we're taking A transpose. Okay? So what I'm asking you is I give you a large matrix. Can you give me a subsample of this matrix which best maintains distances between rows and columns in this matrix? So that's what this is doing. It turns out that to do this, basically, all you have to do is to compute your singular vectors, and you can take some norm of the singular vectors corresponding to the rows, and those norms will give you some sense of how important a row is with respect to defining distances. Okay? So this is a very quick and dirty of leverage score sampling. There's a whole bunch of mathematical proofs that lead up to the fact that the expected value of A transpose A, which is the original distances in the matrix, minus A tilde transpose A tilde, these are the distances in the subsample matrix, that these distances are quite close. Okay? What this tells you is that I can take a large matrix, I can sample a much smaller part of the matrix, provided I'm doing this sampling using this thing called leverage score sampling. And if I did the sampling this way, I'm going to, for the most part, maintain distances in this particular matrix. This is kind of cool because what this gives me is a hammer by which I can find out all the important parts of the matrix. Okay. So how well does this work? Let me set up a simple experiment. If you guys are interested, the go-to data set here is something called the Human Connectome Project. Really, really nice data set, very well curated, extremely large, so you can't just download this data. You have to apply for this data, send them a bunch of money, and they actually send you the disks. They don't want, this is large enough data that they don't want to maintain a site where you're downloading this data. So this has structural, functional MRI, EEGs, MEGs, so about 1,100 subjects. We mostly focus on functional MRIs, and there are different categories in this data set as well. There are twins, there are related subjects. We are mostly looking at unrelated subjects because we don't want to bias our results. Data acquisition is done over 2 days, and on day 1 you do 4 tasks. On day 15, 1 resting state and 3 tasks in your second session. All right, so the first question you're looking to answer is whether there exists a small set of features that code individual level identity. And what we show here is that what we're going to do is to take that, somehow build that large connectome, and then sample that matrix. And we're simply changing the number of samples that we're pulling from that matrix. So these are the number of features. So that's the x-axis. And the right axis is the accuracy of current classification. In other words, like I said, you take the subspace and you're computing the correlation only in the subspace of the markers. And you use this correlation. You pick the one that is a maximum correlation to say that one is you. And you determine the accuracy of what fraction of those correlations correspond to you. And you can see that this very quickly saturates. Up at about 60 features, you are pretty much at 90-95% accuracy, which is about as high as it gets. Okay? And that's 60 out of 64,000 features. And what that tells you is that there's a tiny, tiny fraction of your brain that is unique to you. And all I have to do is to just look at those features, and I will tell you this is who you are. And it turns out those are actually very interesting features, too. So are these features anatomical? So the question is, how do we do anatomical mapping of individual specific signatures? And recall that each feature is actually an entry in the correlation matrix. That was the matrix we were sampling. So basically, every time you pick a feature, that's giving you one edge in this matrix. So in this particular case, I show you one particular sample of the edges that I pulled out, and you can tell that the leverage score regions are very spatially localized. And in fact, we'll show that these features do actually end up with things like cognitive processing, which are strongly known to be unique to individuals. And once again, these are very, very high, low p-values, high statistical significance. We're talking p-values of 10 to the power minus 10 or less here. So now the question you're asking is, can I find markers for task-specific data? And sure enough, we can go through exactly that same procedure and find the markers of task-specific data. And we can show that we can actually, with very high accuracy, tell you exactly what you're doing. For the most part, most of them have very high accuracy: social, working memory, relational, motor, gambling, emotion, and language processing. Except for a couple of topics, motor and working memory, which don't have as good classification. And the reason for that is these are actually complex tasks. When you're doing a motor task, it's not just a motor task. Your visual field is also active. Of course, there's cognition. So these tasks are more complicated than the simpler tasks, so they have slightly lower classification accuracy. The rest of the tasks have excellent accuracy. All right. It turns out that markers of a task code for identity even when subjects perform different tasks. So for example, if you were doing language processing on day 1 and you were doing emotional processing on day k, I can still pick out who you are. So all of these are very, very robust. So let me skip through these a little faster. The one last result I do want to point out here is that the performance itself can also be inferred from these markers. So all I do is I find those markers, and I fit a regression model on top of these markers. And on the test set, I can show that I can get you very high accuracy in predicting how good you are at doing these tasks as well. So these are all very cool. So we got our feet wet. Now let's look to see if we can be even more aggressive here. So let's now look to see if we can predict what you're watching or what you're listening to. So this experiment is a whole bunch of people, as I mentioned, sitting in an fMRI machine watching Sherlock. This is generally called naturalistic stimuli. This is watching a movie, listening to an audiobook, things like that. And as you may imagine, inferences from neuronal response to natural stimuli inform a number of important things about cognitive processes. So for example, you could say, how similar is our neuronal response to the same input? In other words, if we see the same thing twice, do we have the same response? You can ask the question, how similar is our neuronal activity when we recall from memory? In other words, first I'm watching a movie, and then I'm recalling the movie. Do I expect to see the same kind of response in my MRI? We can do sequencing. If we recall, do we recall in the same sequence as we watch? Clearly we don't. So what are the responses that come out of these things? How similar are our responses to the same movie but in different languages? So these are all really cool questions that you can start to answer with these things. All right, so let's start with a few simple objectives here. The goal is to find a representation for a given fMRI frame that is consistent across subjects. In other words, if you and I are watching the same clip, we should have the same response. We want these representations to also capture frame similarity. In other words, if I was watching two frames that are similar, then my response should be the same. So it should be the same within an individual. It should be similar across individuals. And the question is, how do you find such a response? How do you take your terabytes of data and distill it down to a vector which says that this vector corresponds to the response that you expect to see across a group of individuals in response to this movie clip? And the tool that we use here is something called archetypal analysis. So let me give you a quick and dirty of what archetypal analysis is. Think of it this way. Forget the math. I'm a very visual person. I've given you a bunch of points in very high dimensional space, and I've said, OK, find me a convex hull. Just think of this as a polygon, a convex polygon, with k corners, which encapsulates as many points as possible. Okay? So think of this as I don't want to use a computer science term, it's a convex hull. It's an approximate convex hull in very high dimensions, which is a hard problem, but I've given you the number of corners of this convex hull. Okay? So the cool thing about this is, if you were to somehow represent all of these MRIs as points in a very high dimensional space, and if I were to somehow give you this convex hull, the beauty of a convex hull is everybody else sort of sits inside of this convex hull, and the convexity means that each person's response is some scaled sum of all the corners. And the convexity constraint would mean that the coefficient sum to 1. In other words, I could be like two people. I'm 0.8 like this person and 0.2 like this other person. So all the coefficients are positive, and all the coefficients sum to 1, which means that I can really attribute myself very nicely to one of the corners of this convex polytope. So this is what archetypal analysis does for you. There's a mathematical view of this as matrix decomposition, but nevertheless, the visual interpretation of this is quite clean. All right. So how do we use archetypal analysis here? Well, we divide the episode of Sherlock into scenes. So because different scenes will evoke different responses, so we want to separate these out into scenes. We don't do this. Experts have already done this because we don't want to cheat. So we use third-party annotations. It turns out each scene corresponds to a few tenths of seconds. So think of fMRIs as you get one MRI frame roughly about 1.2 seconds. So we're talking a few seconds per scene in Sherlock. We stack all the fMRI frames for a scene from all subjects into a matrix and then do archetypal analysis of this. And now you will notice that archetypes represent snapshots of brains corresponding to the scene. In other words, if you see one dominant response, that can be associated with a scene for which you get that response. I apologize, Lisa, I don't have a clock. And I don't know if this clock is accurate. How much time? 2:58. Okay, I'll take about 10 minutes. Okay. Is that okay? I will try. Okay. Sure. So for each fMRI frame, we find the closest archetype, and we find the consensus archetype for each time frame. We call this the dominant archetype for the frame. Let me skip through this a little bit. This is a pictorial, how I've stacked all the frames together. The responses for all of those frames become a matrix. I have now computed the archetypes for each of those scenes. All right, here's the data set. The data set's got 17 subjects, 50 minutes worth of viewing, about 2,000 fMRI frames, 50 scenes, third-party annotations on these things. We only use regions of interest from hippocampus and the dorsal default mode network. These also come from other domains. And hippocampus has been implicated in episodic memory, and DDMMNs are active for viewing stimuli. Oh, sorry. All right, so here what we're trying to do. We're trying to validate that frames with similar or same archetypes are actually similar. Okay? In other words, if you see two similar archetypes in your fMRI, you should see similar things happening in your scenes as well. And for this, we use third-party annotations. We don't look at annotations on the scenes either. It turns out that we find that pairwise archetypal similarity of frames is a good predictor of the scene similarity as well. And I'll skip quickly to what these little heat maps are showing. The first one is frame-wise similarity of dominant archetypes. This is from the fMRI space. The next one is a frame-wise similarity of expert annotations. And what you'll see from those first two, and you see a product of the first two and the third one, is that scenes that are very close to each other in their fMRI response, as coded in their archetypes, are also very, very similar in the visual part of the frames, as represented by their annotations. So that's really cool. What this says is that things that are close in the visual space are close in the response that I get from my fMRI. Turns out that these are actually also predictive. I'm going to skip through this detailed experiment because I do want to get to the next one. But we can actually also predict what kind of a fMRI response you will get when you're watching a certain scene, or conversely, if I see a certain response in the brain, what are you probably watching? Okay? At least in terms of annotations. So this is really cool. So we've gotten one level cooler in our cool scale. Let me skip this. And now let's get to the really, really cool stuff. So this is truly reading people's brains. So this is reconstructing visual stimulus from MRIs. So we've shown that dynamic neural responses to continuous visual inputs can be expressed as low-rank representations or archetypes. Now I'll show that I can actually reconstruct images to the point where a third-party pre-trained neural network can say something like, aha, you saw a car, you saw a face. And I'll show you that I can do that with quite high accuracy. All right, so what is the technical approach here? We're going to divide the video into, of course, train and test sets. The first thing we'll do is we'll train an autoencoder. So we will train an encoder-decoder deep neural network, which basically will take your images, your video frames, and they will code them into a latent representation. Separately, I'm going to train another map that brings you from your MRI into the same latent space. Okay? And of course, there's a decoder that takes this representation in the latent space and reconstructs the video frame. So these are the three basically neural networks that I have to train. And visually, this is what it looks like. So think of the video frames coming from the top. I've now trained an encoder-decoder, which basically is an identity function. I have pinched the neural network in the middle, which means that I get a latent representation, which is much smaller, only in 1024 dimensions. So I'm going to have a 1024 dimensional representation of each video. And then, of course, the decoder will reconstruct that video back from that latent representation. Separately, I'm going to take the fMRI, and I'm going to train a separate map, which is the fMRI latent space map. And you set up all the loss functions. The encoder-decoder loss function is to simply recreate the video. The fMRI decoder loss function is to go to the original video from the fMRI space. Okay? So I'm going to skip the details of how all of these neural networks look like. I'm happy to share these publications with you, but that's sort of what the encoder looks like. It's got a bunch of convolution layers and pooling layers, and you flatten it out and followed by GRU. The decoder has got convolution transpose and upsampling to get that image back. And that's what the map looks like from the fMRI to the latent space. It's got a couple of GRUs and a dense transform to get the latent vector. All right, since Lisa is going to be angry at me, I'm going to skip a number of these results. And I am going to go straight to, let me go the previous result. Okay. Oh, come on, yeah. So let me start from here. So remember, I can get to the latent space from two places. I can either get to the latent space from the video frames, or I can get to the latent space from your fMRI, okay? So the red dots are the latent embeddings from the video, and the blue dots are the latent embeddings from the fMRI for the same video frames. Notice how close they are. This is really cool. What that says is that in the latent space, I can basically start from your fMRI, and I can get to basically the same point you would have gotten to had you started from the video frame. I don't need the video frame anymore. I can simply come from the fMRI onto that latent space and then use the decoder to reconstruct that video. Okay? So how does that work? All right, so what I've done is, on the left is the latent vector from the visual stimulus. The right frame is the closest neighbor from the latent vectors from the fMRI frames in the training set. So what I've done is I've populated this latent space with a whole bunch of training videos, okay? And all of those videos exist in that latent space. On the right, I have unseen videos, okay? And what I do is I come to the latent space as well, but I'm not using the decoder directly. I'm moving that latent space point to the closest point that corresponds to a video that I've already seen. Okay? And this is what you get. So you see on the top left, well, what I really should have seen was a skyscraper of some sort with a sky, and that's almost exactly what I had seen in the training set. So this is not true reconstruction, but this is kind of like saying, hey, tell me something that looks like this that you've already seen before. And that's what this is showing. And some of these are really cool. I mean, look at the fingers, the people's faces. So this is cool, right? But this is still not reconstruction. This is simply, tell me something like this that you've seen before. Now you can actually do full reconstruction. And that is reconstruction, okay? So yes, that doesn't look very much like a face, but if you squint hard enough, and clearly a third-party neural network can identify it, that's somebody's face that was the original frame, and on the right-hand side is a reconstruction. And yes, this looks very hazy, and you might say, you're cheating at this point, but really the results we show is that we use a third-party trained neural network called MTCNN. This is not ours. We didn't train it or anything. But basically what this neural network says is that if you find a face in the original video frame, with more than a 70% accuracy, you will also, the same neural network will also find a face in the reconstructed frame from just the MRIs. So we've gotten to the point where we can do more than faces. We can do cars, outdoors, indoors. We can classify into about 8 to 9 different classes on these really, really crude reconstructions that we're now building. And you could say, what would it take to make this better? A bigger training set. Remember, we're only training this on about maybe 30, 40 video clips. If you had a much bigger training set, we could probably get a much, much better reconstruction on these things. So we're to a point, basically, where we can probably start to read people's minds on what they're watching. All right, I'm going to stop at this point. A bunch of papers here. I'm happy to share these with you. Most of these should be online as well. If I had more time, I would have a whole bunch more of these, but I will stop at this point. Thank you.